Hello, everyone. I thought before I start my talk, I'll just put a brief plug-in for a conference that we are organizing in 2026. So if you're anywhere in the London area or would like to join us to talk about cytoskeleton, cell division, migration, addition, it's a great opportunity. Okay, so I wanted to speak about opportunities and challenges in deep learning when I first saw the call for abstract. But then I started adding more things, removing more things when I heard all your fantastic talks. So I want to apologize. The title doesn't fit with what I'm saying. However, everything that we do is all that is there. So probably this is actually a better talk for the audience. So, in my talk outline, I thought I'll start with how my journey began into this area of using deep learning extensively in the lab. So, we were cell biologists looking at chromosome capture. Then we got introduced to machine learning tools, and then we started building our own models. And that's how we have now SpinX. And then how we use SpinX to look at spindle rotation mechanisms at a very, very high resolution are going to be towards the end. Everywhere, just to make sure I'm keeping with the title, I thought I will say opportunities, challenges, so at least it's kind of connecting. Some disclosures I have to make. So our work is also co-funded by industry partners. There are two research areas my lab looks at. The primary one is how chromosomes are segregated. And from there was born a second question, how are spindles rotated? They're somewhat united by microtubules and microtubule regulating forces that operate during mitosis. Because it's a little bit of a techniques-oriented session talking about deep learning, I don't develop algorithms, I only develop applications. So I thought I would also say what kind of research approaches we have where deep learning models have made a big difference. So we work at very high resolution movies as the one that you see here. Just want to get the right pointer. So this is a photo inactivation movie. So we shine blue light at a specific time, and then we can create small lesions at these dots, which are kinetochores, whenever and wherever we want. And then we can image at 60 nanometer resolution. Now you can see there are lots of spots, a variety of intensity changes. So manual measurements can be very painful for cell biologists. So we trying to seek image analysis tools there. We also look at this kind of relatively low resolution movies, but has hundreds of events because there's just so many cells dividing under the microscope. This again is better analyzed in an automated fashion. And then out here, what you see are microtubule ends tracked by the growing microtubule-end marker, EB1. And that's in a millisecond regime. Again, millions of particles moving up and down in 3D. We do need particle tracking softwares there. We also have another variety of movie where we are looking at cell fate, DNA damage, and what's impacts on cell fate. And these movies run for 48 hours. We've started doing this on iPSCs that run even half a week. So they obviously are hard to analyze manually, right? So these were the reasons. Okay, sorry, this one. We started looking at tools. So there was a wide need for automated tracking of subcellular structures ever since I started doing microscopy. So for example, the basic biology studies that I showed you, how microtubules rotate the spindle, how microtubules pull at kinetochores, and we also look at very, very few, but we also look at preclinical models, just singular I wouldn't say these are organoids. These are intestinal cysts where you can look at multicellular events. And in here, we have routinely used high resolution imaging. And applied automated image analysis, which is what I'm going to today introduce to you about, and taken some of the data for modeling. A lot of data is just not modeled. I'm always looking for people who will come to my lab and say, we want to model. So there is data there. We have managed to progress through some amount of modeling, but not all of it is modeled. There's another area where we don't actually model or draw out any measurements for looking at microtubule-mediated forces, but just to look at screens that we run for drug target identification here. Although you might say this is like a yes or no study, for example, whether a cell lived, whether the cell passed through G1S, even there, it seems very straightforward, but it's not very straightforward when the data becomes huge. The one part that I don't talk about, but we are actually working very actively because we have funding very actively in that area, is variant stratification. So naturally occurring gene variants across multiple ethnicities can impact the processes we study. And these processes do have a role to play in diseases and in drug resistance. So we are trying to take what we have found in basic biology all the way to understand whether a variant in a human is harmful or harmless. So that part is something I'm not talking about, but if any of you are interested in variants or studying variants, please let me know, because we have a large global partnership trying to pull together our data to build a database. I think I probably will have one slide on it. Okay, to introduce to this audience, Nigel already introduced this very beautifully. There's the kinetochore, a macromolecular structure of 138 proteins, many of them multimerized. So if you look at units, there are over 20,000 units of proteins coming together at this structure to harness the forces from the growth and shrinkage of microtubules to power chromosome movement. We wanted to know how is this large structure tethered to a dynamic object that's continuously falling apart, right? And when I started the lab, it was very clear chromosomes are tethered to microtubule-ends. There was no questions about it. Plenty of EM images very beautifully showed that they are tethered. But even today in human kinetochores, we don't know exactly what is tethering. Right? So the paradigm then was microtubule ends will just grow and shrink stochastically, and they will just bump into the kinetochore straight and capture. That was the paradigm. I mean, I had watched by then several movies. It was not what was happening, but I was a postdoc then. So I thought, okay, this must be a good area because obviously the movies don't show this. So there must be a gap. Unfortunately, at that time, computational models had challenged the view that if you have a 40 nanometer wide structure searching for an object that's around 400 nanometer in this huge volume, especially when it's masked behind chromosomes, it's very unlikely going to find everything in like 5-10 minutes. And they, in fact, predicted, I think, several hours for the capture process to be complete. So the answer was we needed high resolution live imaging. So when I set up the lab, I thought we are going to devise microscopes in a way that we can actually monitor this process in live. To cut a long story short, it led us to talk about this end-on conversion process, which I'm going to just briefly introduce in the next 2 slides. So what did we need? We needed simultaneous visualization of kinetochores and K fibers. I'm sorry, the leftmost is getting cut off, but I'll read out when I need. So we wanted to not go back and forth between a green channel, red channel, FITC, TRITC. We wanted it to be simultaneous. So we wanted to look at dual mount cameras and then make our optics in a way that there is no lag between the 2 colors. So that's what we managed to build together with INSOL and Applied Precision. And at that time, there was also another engineering team, Cambridge Sense, which helped us. In here, just to make sure it's easier for you to see, I've put the stills there. What you can see is a bundle of microtubules very close to a centromeric marker. So the kinetochore builds on a region of DNA called centromere. So that's the red one. I'll soon tell you how this became a savior for us. And we would search for these kind of detached kinetochores in our movies manually. Roshan was perfect in that. Actually, Roshan is now running his own lab in NIH. He says he's not anymore searching in basic biology. He's doing the same in pathology, he says. So then we look for kinetochores that are captured along the walls. We keep tracking them to a point where we can see this black gap. So this darkness was a joy for us because that means that's the kinetochore, which we are not seeing because we have not labeled. And then the microtubule tip has tethered to the centromere. So once this event had happened, then we could actually go back to the history and say, what were the previous steps? Now, we did this for many, many years. And this is a process we called end-on conversion, similar to what was reported by Tomo Tanaka's lab in East. And this was the first time human chromosome capture was visualized in this manner. And this is a completely untreated cell. It's just expressing tubulin and a centromere marker. But you will soon see why this could not be scaled. You know, even though we had Roshan, who was terrific, we could not scale this because we were chasing for these few events, right? In that entire movie, that green was what I was chasing for. I'll soon tell you what is Astron and what is MAT2, but the number of events we were chasing were very, very few. So that we recognized that it was a challenge. So we decided to treat cells with monastrol, which is what you see here. Then you arrest your cells in monopolar spindles. So you're not allowing them to ever form bi-oriented attachment, which was like a brief 5 to 10 minutes. So we had this plenty of time to now watch the event. So what I'm showing you here is an end-on tethered kinetochore. So in blue is Astron. So before this study, Astron was known as a kinetochore binding protein. It was also known that it binds to ends of microtubules. What was not known is that Astron would bind like this to monopolar kinetochores. People thought it is something that's coming because of bi-orientation. So in fact, when we tried publishing this, there was a lot of resistance to this view that you can actually have Astron arrive at such an early stage, even when the other kinetochore is not attached. I'll show you more movies and pictures so you will see what's happening. MAT2 is a checkpoint protein. This was a very well-known protein because it's conserved all the way from budding yeast to humans. And this protein is supposed to leave the kinetochore as soon as the attachment is right. But even then, the idea was that the attachment is correct only when you make it bi-oriented. That is, the two sisters are connected from opposite poles of the spindle pole. Right. So we were studying in monopolar spindles, and people were like, "What's happening? Why are you losing MAT2?" But well, now we know. Now we know this is what happens as soon as end-on attachments are formed. MAT2 leaves, Astron starts arriving. Okay, so the end-on conversion gave us a lot of material to work on. I'll give just a brief summary, I think, next. The process, it's a complex process because kinetochores captured along walls have to be brought to ends without detachment. So you can imagine a large structure like the chromosome now becomes attached to the microtubule ends, and there's no detachment in between. And only microtubule-end attached kinetochores recruited Astron's cap, and that was independent of bi-orientation. So we had a probe. We could hold on to this tool and say, how is this process regulated? So that's, I'm not going to go into all of the papers, but we could then assign steps of the process because we would just simply do RNAi of a variety of kinetochore proteins and ask, where are we stuck? So we could see that step one obviously was microtubule wall tethering. If you had huge lesions on the kinetochore, like didn't have a very well-known kinetochore binding protein called microtubule binding protein called the NDC80, or its platform, MIS-12, then obviously even this event wouldn't happen. But once this event happened, we needed kinesins like SEMP-E to bring the kinetochore closer to the tip of the microtubule. Now remember, these are monopolar spindles. In bipolar spindles, obviously these kinetochores would arrive to the tip in a SEMP-E independent manner as well. And then once at the tip, we needed the loop domain of NDC80 for the kinetochore to remain tethered to the very tip of the microtubule. In the absence of NDC80 loop domain, the kinetochore would simply remain laterally tethered. There was also one more interesting observation that we had was even when you have all these players, if microtubules were not destabilizing normally, so when we remove a kinesins like MCAC, then the kinetochore struggled to form end-on attachment. So it formed end-on attachment, very quickly lost it and became laterally attached. So there are all these really, what do you say, resilient researchers who analyze this painfully manually, right? I mean, these are like fabulous PhD students who worked on these papers. Oh yeah, so those are the steps. Step 2 is kinetochore moves towards microtubule-end, and step 3 is shrinkage, and the step 4 is end-on tethering stably. Then it remains stably. So this is all old work, but that meant, as I told you, we could find that you had SpinX² as a complex, not only arriving at the very end, but also you needed this complex for keeping the kinetochore at the end. So now after these folks left the lab, they went to postdocs, became group leaders. We were still collecting movies of this kind and trying to look at regulators of SpinX² because we knew without SpinX², you can never form those end-tethered kinetochores. So this was actually started off by Duccio and Senong. What they did was they started watching Astrin very carefully at end-tethered kinetochores. And what they could see was, so you remember there was a lot of resistance because when Astrin was discovered in 2001, it was correctly reported that it's present only on congressed kinetochores. Because you remember my first graph, it's only in 2% of the cases you can actually capture lateral attachments if you are in metaphase. This is just really few kinetochores. So they just quickly saw and they said, oh, it's only on congressed kinetochores. But if you do not let kinetochores to congress, trap them in monosterol, you started seeing these kind of beautiful astral decorations. So the green signal is on the sister that's facing the pole, and there is no green signal. So the NAF2 is here, is a kinetochore marker. There's no green signal on the kinetochore that's unattached, right? So the attached kinetochore alone brings astrin. And this, the way it brings was also very beautiful. So we spent a lot of time enjoying this in life. So astrin first builds like a nice trajectory along the K fiber. The comet just extends further and further, and at some point it becomes like a boom present. We don't know what makes this transition yet, but we know kinases and phosphatases and mutations that can either give you just the crescent or the sleeve. Okay, where are we now? Okay, so this is one of the slides during that search for regulators. It's actually for Radhika, for companies. This is my only Aurora B slide today. So what we found was that if we brought Aurora B kinase to the outer kinetochore stably, then you can never form end-on attachments. This was also a surprise because Aurora B is thought to be an error correction enzyme. So this is a kinase that's supposed to detach kinetochores from microtubules. What we could not see was any detachment. What we saw was that you can see kinetochores nicely decorating the walls. That's what we could see. But these kinetochores that were decorating the walls did not have astronaut SCOP complex. And we could reverse the process. So this is not because we brought, so the trick to bring Aurora B is MIS-12, and then you make a fusion of an Aurora B platform Mis12 is a kinetochore platform, kinetochore protein. So this is a way you could increase Aurora B, but we know that it's not because of this trick, because if you inhibit Aurora B in these cells, you could now form very beautiful end-on attachment. It's almost like these experiments were really easy, like, on-off switches. So we know that Astrin's cap is counteracted by Aurora B kinase. And in this process, we were also looking at other kinases, obviously, as controls, but then they became a story by themselves. Another kinase that counteracts is cyclin B-CDK1 kinase. And this was, again, a big surprise for us because cyclin B-CDK1 is very high in the cell during pro-metaphase when kinetochores are captured. However, cyclin B-CDK1 leaves the kinetochore as soon, in this paper, we show that it leaves the kinetochore as soon as end-on attachments form. So this is a DMSO control, and you can see these astrin, the beautiful sleeves of astrin forming the crescent, and then it will quickly go back to its sleeve, and then it'll grow back into its crescent. So it's always oscillating back and forth. And then you can also see the other kinetochore has no purple signal whatsoever. That's because it's unattached. However, when you add the CDK1 inhibitor, the unattached still remains free of the purple signal. So it's not that CDK1 inhibition just will bring astrin, come what may. You need the microtubule as well. And the microtubule is now, when it's end attached, you have astrin collecting as a nice marker. So why did I go through all of those details? The reason I went through all of these details to bring you to this cartoon, which has a variety of names. I don't want you to look at any of this, but each one of them has a story with a movie of that kind. So there was just too much data in the lab with all of them willing to do manual analysis, but there was a lot of data coming in. The story ends here that we finally now know you need only 271 amino acid of astrin to recognize this end-on attachment status. So there is something happening at the kinetochore, and this recognition happens. Okay, so that brings to my transition slide. So we had terabytes of really high quality data. Terabytes is an understatement. I think we had maybe nearly petabytes, because at some point Cambridge said, we have to tape all your data down because we can't store it like this. And we were like, oh my God, we don't know how to get it out of tapes after a point. What are you talking about? And then, we have put so much effort. What can we do with it? Maybe there are some subtle phenotypes that we didn't pick because we are doing some experiments hurriedly for revision, and maybe they are key. Maybe there are other subcellular structures we want to see. Maybe we want to look at the entire spindle. Maybe we want to look at the spindle pole. So then came the need. So then we thought, okay, why don't we just get another grant written to see whether they will let us study not just these kinetochores, which we were focusing on, but this entire structure, the spindle. We can just say, look, the spindle is a large structure. We don't know how it moves. We are going to study it. And luckily, we did get funded. But then when we realized, that also meant we had to do a few more new experiments, because it's not just a spindle that's needed. You also need to look at the microtubules of the spindle. So we started making high resolution movies of the microtubules. So to give you a brief introduction here, before I go into where we came with the deep learning model, why we came to the deep learning model. So the current model, but also at that time, the model has been that there is dynein at the cell cortex, and it pulls at microtubules that reach the cortex. But you can see this movie, and you will see the comets. Sorry, I think I should not use this. I should use this. Can we? Go. So if you see the number of comets that reach the cortex, are very few. Actually, there's just 4 or 5. You see 1, 2, 3, 4, 5 comets. In this particular line, there are very few comets. So we had to make a ton of movies again to pick up how the spindle is rotating. So there were even more movies kept forming. And we had already told our funders, we're going to look at what are the forces that counteract these pulling forces. So we needed to make screens like RNAi, large RNAi screens. So then the thought was, let's just do automated tracking. At this time, this is 2009, 10, there was no there was machine learning, but not for image processing as of now. So we went through the traditional way of just segmenting the images and trying to put it together. There was a lot of manual correction that was needed. Adam, who now leads the image analysis for AstraZeneca Cambridge, he was involved in, he was the one who was driving this project. We could learn a lot from the movement, but also he managed to model some of the key forces that we will have to consider for the spindle to move. But this process was very helpful because this told me, even if you had an automated tracking system, many of you probably know it. I didn't know it at that time. You have to correct. If you really want to trust the data, you have to sit there and manually still double check. So the students were again doing it manually, right? So all these movies we were trying to do. So the limitations were we needed manual adjustments based on how bright your image is, how dull your image is, whether the spindles were near each other, or whether they were in focus. And you could capture prominent defects, like, okay, the spindle is really bad, it's moving very badly, but you could not still use it for very precise 3D measurements because of this time gap. Sometimes T1 to T4 was picked and then 4 to 6 was not picked. So we were facing problems. So, and we were looking at solutions. So we had not stopped. We were looking at solutions. Deep learning had not arrived. The word deep learning had not arrived, but there were these KNN neural networks. And David Dank is amazing. Actually, he's now leading Deloitte's AI global team, consultancy team for the entire Deloitte. So he decided we should bring KNN to this because it would be, you can just go dynamics blind and it will segment very easily. So it was still supervised machine learning, but it was very suitable for image classification. And then we were able to pull out these spindles very, very well, very clearly. And then we could pull out chromosomes, cell cortex, anything that you said you wanted, you just had to label a few images and it was pulling out. This was like revolutionary. So everybody in the lab wanted something like this for themselves. The people who studied kinetochore started marking kinetochore. Those who studied nucleus started marking nucleus. Spindle poles, so everyone could then move forward at their own pace based on how quickly they could annotate a set of data. And for the spindle alone, because our movies were ancient, right? This was happening in 2014. Our movies were from 2009. So the resolution was low. We had only 3 or 4 slices, not the beautiful reconstructions you get in our light sheet today, but we still wanted to make use of the old movies. So what David also did was try to put together 3D modeling, considering that fact, two features, that the spindle will always be constrained within the cell, within the cortex. And then the second one is the spindle poles are the highest intensity. So if you had a spindle tilt, you could still be able to mathematically model how, in which Z plane the spindle was. So that gave us SpinX. And I've just put one example movie here. But with something like this, it really paid off. We can look at hundreds and hundreds of cells. For every condition, we just easily make hundreds of cells and run through SpinX. And you can see how it tracks. So it tracks both poles. It can see whether it's a full tumble or the poles are migrating in a way. So what we are measuring here is the pole to the cortex distance. So the poles are going very close to the cortex or away from the cortex, and you can see this through time. So SpinX came at a time was also luckily the time when Zeiss wanted deep learning tools, and they didn't have many. So now SpinX is available in Zeiss as part of our IBIS Pro. To implement SpinX on Zeiss, Bingao helped us a lot. So Bingao is here in the audience. So Bingao knows the pain of working with biological data. He's an AI expert. He spent a lot of time in the lab trying to get us into SpinX. What was the advantage of SpinX? Like, in addition to all the story that I told you, so I'm a cell biologist. I wanted to run a lab efficiently. I could see the pain of all those hardworking students. So I was very grateful that there was a spindle tracker. But the beauty of it was now we could track Astron movies. Astron is a microtubule binding protein, and it decorates the entire spindle. We could track EB3 movies. The EB3, dots that I told you, the dots cannot be tracked, but the entire spindle could be tracked. We could also modulate colors. So, MK2 EB3 usually looks slightly different from MRFP EB3, but those things didn't matter for SpinX. It would just track it. We could track dyes like sertubulin, which didn't really label the astral microtubules. In addition, SpinX would also track very beautifully. This is actually data set from a public database. It could track mouse ESC cells, HEC293 cells, bovine cells. So these are not from our microscope. This is from somebody else's microscope. So it's working really well across sizes, across different microscopes. So now we said, okay, let's come back to biology. So I'm now switching back to why we built SpinX. We wanted to know, can we use the SpinX DL model actually have a deeper analysis on spindle rotation mechanisms? Because previously we were just saying, it's a spindle aligning along the long axis, because it's very well known that the interface long axis is remembered by the cell through retraction fibers, and the spindle is always aligning along the long axis, so the two daughters can form parallel to the long axis of the cell. Okay, so this slide basically is, we do cell culture just as before, make the time-lapse movies. Immediately the movies are sent to SpinX or MultiSpinX. This is Bingao's contribution. Actually, SpinX, we had to crop it for a single cell, but then MultiSpinX meant you can have multiple cells, and there was a way to crop automatically as well. And then we would just do statistical analysis. So this is the outcome of our screen. So here I am just showing you the cartoon of what I showed you earlier. So there is the LGN platform that brings dynein, and that dynein is thought to pull at the spindle. So so when you remove LGN, because you've removed the platform and there is no dynein, the spindles don't move at all. They remain centered. They are where they are born, and they are in the center of the cell. So there was no surprise there. We were happy that it was as it is expected. You can remove another molecule. This is a polarity molecule called PAR3-2. Again, the spindles remain centered. But when we removed PAR1, which in humans is called MARC2, we were very surprised. The spindles started off-centering. Right? They were never in the center. Now, MARC2 is a kinase, so its RNAi has to be done very carefully to see. I'll show you some inhibitor treatments as well. But when this off-centering was happening, because we had this whole data set, there are some cells where the RNAi is partial, so we could actually pull out all of this data to ask what was happening. These spindles that could not center, we finally, in the paper, we say there is no cortical pushing. But at that time, we did not know about pushing. We just could say spindles off center in the absence of PAR1 kinase. And this was really nice because PAR1 is a polarity kinase. It's known to play a role in C. elegans embryos, but its role in human cells was not known. This movie, I put this movie in so that you can see actually how difficult it would have been without something like SpinX to see this. So if you see in anaphase, so the movie all the way goes to anaphase. That's Mark II RNAi. Yeah. So if you see in anaphase, you would see these spindles are centered. So if you just had a quick look at the movie, you would have said, "Oh, these are centered." But because in prometaphase and in metaphase, the spindle makes these very nice gliding motions that you can measure very carefully on SpinX. You could actually see that this is gliding along the cortex, whereas the control is not gliding, it's oscillating. So if you see the movements here, it's usually a full oscillation, left, right, but whereas here, it would just glide and even push against the cortex. So even our cortical tool was showing these huge changes in the cortex, pushing against the cortex. Okay, and I'm not going to show you here, but in this paper, we also show there are curly, long microtubules at the cortex when you don't have Mark II in the cell. Now we have a different problem. I put this one slide to just illustrate the new problem we have. The problem we now have is we can measure everything. That means we end up having too many graphs. Everybody brings a graph, and then we put some statistical tool, 4 stars, 3 stars. It's just too many graphs. Now, I don't know whether deep learning will solve this problem, but this is a new problem. But it has helped us to pick certain phenotypes that we couldn't have picked. That is SENP-E. Sorry, it's getting cut. So when you inhibit SENP-E, that's a kinase. And if you remember, I'm going to come one full circle. I'm closing my talk very soon. When you inhibit SENP-E, you have these uncongruous chromosomes. So we had these movies from long time back, from 2009. Like this paper was out in 2023. So we can go back to our data that's almost 15 years old. And the whole spindle tumbles, which you can see very nicely here, how a tumbled spindle comes back to position. You can analyze it. And this is a Mark II inhibitor treated cell. So there now no RNAi. The inhibitor treated cell fails to center. So those are centering in alpha, beta, gamma. You can look at every coordinate that you want to look at. Which is I think is very powerful, because we do see certain phenotypes associated with certain depletions and not with others. But I feel all of this numerical data should go into a model to make more meaningful sense. That's what I would like as the next step. How is Mark II kinase regulated in space and time? I think there are 2 more slides to show you one more challenge we have in this project. Mark II is a kinase, the polarity kinase, that goes to these retraction fibers. So retraction fibers are structures. These are my only contribution to actin in this talk, actually. Retraction fibers have a ton of actin. And Mark II loves to go to those actin-rich retraction fibers. And retraction fibers are thought to be the structures that retain the memory of interface and that guide the spindle rotation. So you're like, okay, so we have a tool now. So let me put the part that you should be looking at. So if you focus only here, there is actin, there is a faint Mark II there, and that's because Mark II is like puncta, not continuously on the membrane. You could maybe see it here better. It's like tiny dots all along the membrane. Mark II kinase, dead. So if you don't have the kinase activity, then it does not go to the retraction fibers. So that's the kinase dead. If you look at how long is Mark II along the retraction fibers, so there's one retraction fiber here with arrows, you can see. Whereas when you have the kinase dead, Mark II now goes only to the substrate, so where the cell is seated on the glass slide. So to reach the retraction fibers, it needs kinase activity. So now we are trying to look at how do these astral microtubules reach the retraction fibers. This is where we have not been able to really watch using the current state-of-art lattice system. We see a few microtubules reaching the cortex, but not many. Partly because when we are imaging somewhere, a microtubule comet just shoots into another plane. So Noriko was a visiting postdoc. What she said was, let's just bring electrically tunable lenses. So the only difference is that these lenses with these lenses, you don't have to move your stage or your lenses, objectives anymore. You can just stay where you are, and we can have quick sweeps, three-dimensional sweeps of the whole place. So I'll show you how that sweep looks like. Chris set this up for us. So the liquid lens sweeps look like this. You capture almost every dot. Sometimes the sweep is so fast, the same dot can be seen coming along with you during your sweep. So you keep watching it. So these movies are really beautiful. But now we have to put together a DL model for learning from these movies, because these movies need image correction. So this is one of the reasons why cell biologists did not use this, although these lenses are used in the defense quite well. And also in the cell phone industry uses it sometimes, but it requires image correction. We are trying to use DL models for correction and also for tracking these comets through 3D. That's my last one. So to summarize, I told you how we showed end-on conversion is monitored independent of biorientation. So the act of making a microtubule-end attachment to the kinetochore does not require biorientation. And I showed you about aspirin. I didn't tell you about the delivery of PP1. So the reason aspirin's cap, after aspirin stop arrives at the kinetochore, several phosphosubstrates are reversed, is because aspirin itself brings the PP1 phosphatase to the end-on attachment and stabilizes the attachment. I also told you that the kinase, CDK1 kinase, promotes end-on conversion. I didn't tell you anything about this work. This is very unfortunate because 80% of my lab is currently collecting variants, naturally occurring variants in healthy humans, in cancer tissues, but from chromosome segregation genes to understand what is its impact in cells. There's a QR code. If you want to go and see whether your favorite gene is in our database, you could, and then you could see whether we are doing anything with it or we have decided to not do anything with it. I told you a little bit about MARC2 kinase, PAR1, which is essential for spindle positioning, and we think pushing forces at the cortex. I introduced you to SpinX, which is a deep learning model that we built, which has really changed the way, at least my lab does, spindle studies these days. We are implementing ETLs. We had kind of abandoned this project, but we now, because there is deep learning is doing so well, we are going to use it to see whether we can correct the comments that we have. Thank you for your attention. I should thank the funders, and I should thank all the people involved in the projects that I just spoke about, and the current students who are working in. We also have a couple of postdoctoral positions. So if you're interested in the kind of work we do, it's for interdisciplinary project. So you have to be able to code and also be on the microscope. It's kind of hard to find those kind of people. Yeah, thank you.
We take a question or two. Yeah.
So it's beautiful talk. So I wonder what brings aspirin to the kinetochore.
Good question. So it's a paper in writing. So we have published that only a 200 amino acid region can go there. So we did some mass spec and we made some structure. So to precisely answer, I think it looks for microtubules that are ordered to arrive. But we really need cryo-EM type of data to nail it down, because at the cell biology resolution, we can just go all the way, maybe 60 nanometer. We do some FRAP and we do some FRET. All evidences are indirect. But we think it looks at it's not looking for bundle microtubules, but a somewhat ordered set of microtubules, then it arrives.
And you mentioned this comet versus crescents. So
yeah.
When exactly do those happen?
So those comets and crescents, I think to be even a comet, it needs that ordered arrangement. But to become from a comet to a crescent, we can only regulate using kinase inhibitors or overexpressing phosphatases. So if we brought
crescent is not nascent too. Does not happening. regular wild type?
No, no, it happens in regular wild. It happens in unperturbed systems. But it was your question, why does it happen? Yeah, so when Aurora B levels drop down, the kinase levels drop down, along with it, CDK1 leaves the kinetochore, then it becomes a crescent.
Let's do one more. Yeah.
So the end-on attachments that you mentioned, I wonder, like all these microtubules, they are like directly connected to the spindle pole, or like they are like branched, and then you have one end-on attachment, and then how does it end up like to a centrosomal connection?
I did ask the first speaker or the second speaker today whether they can see kinetochores. We don't study at that resolution. So we don't know whether it connects all the way. In the literature, it used to be it connects all the way, but now people understand it's broken.
Picture is like it connects all the way. Yeah.
Yeah. Well, our pictures, I don't think you can go anything by that because they are looking at K fibers, not individual microtubules. It's a bundle of microtubules.
I have a follow-up question regarding that. So the conversion to end-on attachments. So one of the pathways, if I understood correctly, you have something that drags the, like first a lateral connection is established, and then something drags those kinetochores towards the plus end of the microtubule. Right?
Yeah.
So I was just wondering, like, if it is also possible that there is a lateral connection, and then this microtubule shrinks and makes a like.
Yes.
Makes it an end.
Yeah, and then the end-on push happens. They both happen. In fact, there are two Nature Communication papers from Eva Tolik. They are, I mean, now you're able to really track them. They didn't, I don't think they did in monopolar spindles. They do in bipolar spindles, I think, if I'm not wrong. But you can see there are two types of movement. One, you remain lateral for a long time. But you have local opportunity to convert it to end on, not close to the pole, but as you move away from the pole, and then there is a push as well.